Great. So I am going to introduce the speaker for the cell biology lab, which is Sharon Batula, and she's going to be talking about the work that she has done this semester studying DNA damage repair in tetrahymena cells. And so Sharon, I will turn it over to you now.
Thank you, Dr. Moore. I'm going to go ahead and share my screen. Can everyone see my screen?
Yep.
Great. Great, thank you. Just want to double check because Zoom's always a little unreliable at times. So hello, everyone. Thank you for joining me this evening. My name is Sharon Batula, and I will be speaking as a representative from MCDB 3140 about my project, which found that the expression of gene 00439330 increases in response to DNA damage in tetrahymena thermophila. Did the slide move for you guys? Oh, there we go, perfect. So just some context, as we may already know, DNA repair, which is the ability of a cell to recognize damage in its DNA and then fix it, is obviously crucial for normal, healthy cell functioning. If errors are left unrepaired, this could lead to a host of negative outcomes in the cell, one of which is cell death. Normally, of course, cell death in lieu of DNA repair isn't ideal, but in the context of cancer, this is actually a desirable outcome. In fact, commonly used treatment strategies like chemotherapy and radiation are designed to kill cancer cells through inducing double-stranded breaks in DNA. DDR refers to the DNA damage response, which identifies and repairs these DNA lesions. And if DDR can be inhibited in cancer cells, then these cells will die more easily in response to treatment because the treatment is inducing DNA damage. As a result, DDR inhibition is currently being studied as a way to sensitize cancer cells to therapy. And this approach would not only enable treatment to be more effective, but it actually may also enhance clinical outcomes through lessening side effects. Because if cancer cells are more vulnerable to treatment, then less of that treatment would be needed to produce the same results. With this implication in mind, for us to be able to target and inhibit DDR, we need a clear understanding of the proteins and pathways that are involved in DNA repair. And as of now, a lot of this information remains unknown and requires further research. So this study sought to explore potential DNA repair proteins in tetrahymena thermophila. These organisms were used as the model organism to learn from, primarily because they regularly induce double-stranded breaks in their own genome and then repair these lesions as a part of meiosis. Additionally, there's a lot of bioinformatic data available for us to reference. And as a bonus, these organisms are relatively cheap and easy to maintain. Gene 00439330, which I'll just be referring to as gene 330 from now on, was pursued as an interesting subject of investigation because of its gene expression profile. So gene 330 codes for a transmembrane protein that's involved in vesicle docking and fusion. And it's been observed to be upregulated at a few different time points during cell conjugation. As you can see on the right, I've circled those for clarity. And these time points of increased expression correlate to known time periods of DNA damage and repair in the process of meiotic recombination. So we wanted to know if this expression profile was connected to the idea of DNA repair, with the hypothesis that cells with DNA damage would have a greater expression of gene 330 than cells without DNA damage. Now, in order to determine if this gene may play a role in DNA repair, we first obviously needed a way to identify and measure this gene's expression. So this is where PCR primer design comes into play. We used an online program to design primers specific to our gene of interest. So I designed primers to identify gene 330. And here's just a snapshot to give some context about the gene. The total gene length is about 1,000 base pairs, but I designed my primers so that they would anneal to about 400 and 800 base pairs into the sequence, which would capture a length of about 400 base pairs. And then if you take into account that there's an intron in this sequence that's about 100 base pairs long, that would get shortened to about 300 base pairs. So now that there's some context about the primer design for this gene, I want to talk about how those were used in the overall methodology in wet lab. So first, tetrahymena cells were grown in culture and then separated into two groups. One group was untreated, left alone, and then the other group was treated with hydroxyurea, which is a chemical known to induce double-stranded breaks. Then mRNA was extracted from both of these groups and then reverse transcribed into complementary DNA or cDNA, because cDNA can be amplified using PCR. For those unfamiliar with PCR, there is a nice image over here at the top. And the idea is you take a template strand, you denature it into its single strands. And then if you insert primers that can attach at a specific point in that sequence, that starts the building block process for other nucleotides to complementary base pair. And through that hybridization process, you can create a lot of identical copies in a relatively short amount of time. And something important to emphasize is that amplification is proportional, meaning that if there were more copies of the gene to start out with, then you would get more PCR product at the end. And how do we know how much PCR product for cDNA is generated? That's where gel electrophoresis comes in. So with gel electrophoresis, when you visualize those bands, a thicker and brighter band is indicative of more cDNA. Now, this assay used to test the hypothesis obviously relies on the primers that I designed being able to anneal and amplify gene 330. But how could we be sure that these primers would actually work in the lab? So that's the question that we wanted to answer for the first experiment. As you can see, here is a gel from experiment 1. On the very left, we have a DNA ladder to give us some reference points for DNA length. And then the first couple of lanes after that serve as our negative control because they were loaded with water instead of a DNA template to verify that there's no contamination of sorts and that no amplification would be visualized. RPPO is a gene that we have primers for, and we know these primers are functional and work in PCR. So this was used as a positive control. And then last but not least, we have the gene-specific primers I designed. And as a reminder, we wanted to see about 400 base pairs and 300 base pairs respectively for gDNA and cDNA of gene 330. And the bands that we visualized match these expected results, which indicated that these primers were functional, effective, and could be used to measure gene expression of gene 330. So with the primers validated, we were able to run an experiment to measure the difference in gene expression between minus cDNA and plus cDNA. As a reminder, minus cDNA came from cells that were not treated with anything, so normal cells from Tetrahymena, and plus cDNA was collected from cells that had been given a DNA damaging agent like hydroxyurea. And here are the results of that experiment. Once again, we have a ladder and negative controls for the first few lanes. Now, something important to note is that there are 3 different primers used in this gel. And RAD51 is the new primer that wasn't in the previous gel. So RAD51 is a gene that increases in expression in response to DNA damage, and it's known to do that. So we wanted to verify that DNA damage had actually been induced in our plus cDNA samples, so we used these primers. And the fact that there is a thicker, brighter band from plus cDNA does verify that for us. RPPO was once again used as a positive control as well as a loading control. And then here we have the results of interest for gene 330. Now for gene 330, it was observed that the bands were brighter at around 300 base pairs from the plus cDNA sample than the minus cDNA sample, which does indicate that the expression of gene 330 increases after DNA damage. And then interestingly, there was also an unexpected band visualized in this last lane that I boxed in green for clarity. Now, the brightness of the bands produced by my gene-specific primers was quantified using an image analysis software. And it was found that the band at 300 base pairs produced by the primers was about 8.5 times brighter with plus cDNA than minus cDNA, which suggests that this gene's expression increases by a considerable amount in response to DNA damage. And then also another interesting point is that unexpected second band was nearly 20 times brighter than the band from the minus cDNA sample. And also this band was located at about 400 base pairs, which does correspond to the gDNA length of gene 330 based on my designed primers. So at first glance, you might chalk that up to gDNA contamination. But if that were the case, we would have seen a band from the gDNA for RPPO as well, and we didn't see that visualized. So with that in mind, there might have been 2 bands due to alternative splicing or 2 different mRNA sequences of the gene being expressed in response to DNA damage. So now, what do these results mean? Well, the most straightforward interpretation of the data is that the expression of gene 330 does increase in response to DNA damage. But also there's the exciting possibility that perhaps a substantial amount of alternative splicing of gene 330 occurs in response to DNA damage. And if that's the case, it's not to a negligible degree either, because in fact, there's about 2.5 times more of this unexpected alternative mRNA sequence than the mRNA sequence we expected. This is especially interesting because the presence or retention of that intron may produce a completely different protein, and this protein isn't at all generated in the absence of DNA damage. Overall, these results are compelling because they imply that this gene, which has thus far remained relatively unstudied, may have a valuable role in the context of DNA repair in Tetrahymenothromophila. And if more can be understood about how this gene functions and interacts with other proteins in response to DNA damage, we may be able to identify molecules that can act as druggable targets to impede DDR. And because DDR inhibition has the potential to increase the efficacy of cytotoxic therapies while lessening side effects, this line of research has massive implications for hundreds of thousands of cancer patients in the future. To expand on these findings, an assay that measures protein activity rather than mRNA expression should be used to determine if a difference in protein activity for gene 330 is similar to the observed difference in mRNA levels that we visualized in this experiment. This may also help to clarify the significance of gene 330 in DNA repair, since as we all know, increased transcription doesn't necessarily translate to a similar increase in protein function or activity. Additionally, it might be valuable to examine the two different proteins that are generated by gene 330 after DNA damage, and gain an understanding of how their roles and relationships with downstream molecules might vary. Ultimately, though, there are a lot of different directions to go from here because the study does warrant further exploration of gene 330 in the context of DNA repair in Tetrahymenothromophila. And I just want to spend a little bit of time thanking some people. First of all, thank you to Dr. Moore for being such a great professor this semester and being a constant source of support and guidance throughout this whole process. I also want to thank my lab TAs, Elise and Kelly, for always being there to help out with experiments, answer questions, and for just generally making lab fun, enjoyable learning environment for all of us. And last but not least, thank you to CU Boulder for offering this class and funding our research endeavors. Also, of course, thank you to you all for showing up and listening. And at this point in time, if there are any questions, I'd be more than happy to address those. Thank you.
All right, well, I'm not seeing any questions in the chat, but you're getting a lot of positive press here. If questions do come up, feel free to shoot them in there or feel free to unmute as well if anything comes up. And I believe that we have one more speaker after this. Is this true, Pam?
That is correct. We are very fortunate to have a guest speaker at this CURE symposium. Our guest speaker this year is, I'm sorry, are there questions in the, I'm worried that I'm skipping over questions because I'm excited.
Oh, we just got one.
Okay, go ahead.
Okay, so this is from Sabrina. She says she also studied the same gene and experienced alternative splicing as well. And in her presentation, she speculated on functions, and she was wondering what your thoughts might be for any speculated functions.
Yeah, great question. It was definitely unexpected because at first glance, it was like, oh, it's a transmembrane protein. I don't know how much of a role this might play. And then it ended up having that alternative expression that was really interesting. I, as of right now, obviously don't know the answer to what role that might be serving in the DNA repair context, but it could be indicative of more of a certain molecule being generated in response to DNA damage and that being shuttled across the endomembrane system more in response, perhaps. I think it would definitely be an interesting question to further pursue because, as I mentioned, there's a lot that we still don't know about these genes, and the more we can learn and understand, the more effectively we can use that information to treat patients and have real clinical applications for that research. But great question, thank you.
And a note from Colin, maybe a new domain on one of the sides of the protein?
Yeah, it's definitely possible. When we looked at our bioinformatic data for this gene, it was like notably very limited. There was not a lot documented. So the only information I had was that domain that is associated with vesicle docking and fusion. But that's a really good point. Maybe this is just another function altogether of the gene that we don't know about in tetrahymena. So that's a great point. Thank you.
Any other last questions for Sharon? All right, well, with that, I'm going to applaud again. Great job.
Thank you, student speakers. The talks were absolutely fantastic. I think if we were all in a room together, we'd all be cheering for you. I think you represented the courses extremely well.